Good morning, everybody. Okay. It's my pleasure this morning on behalf of the Center of Integrative Environmental Health Sciences and the Department of Pharmacology and Toxicology to welcome you to our Environmental Health Science Series seminar. And with me this morning, we have here Dr. Liqing He. Please permit me to read bio. Dr. Liqing He is an assistant professor in the Department of Chemistry, University of Louisville. She earned her PhD in molecular biology and biochemistry in 2016 and joined Dr. Zhang Shan's lab in Department of Chemistry the same year. Her research focuses on developing advanced chromatography mass spectrometry-based analytical platforms for metabolomics and epitranscriptomics. Dr. He has developed quantitative methods for analyzing short-chain fatty acids, bile acids, nucleosides and nucleotides, and oligonucleotides, and has applied these methods to complex biological samples, resulting in 41 publications since 2018. She has received one R21 grant as the principal investigator and 2 external grants as a co-investigator. Our current research aims to investigate RNA modification dynamics and metabolic regulation in alcohol-associated liver disease. Today, she will introduce methods for detecting and identifying RNA modifications. Please welcome Dr. He to the podium.
Thanks, everyone. Thanks for your introduction. I also want to say thanks to Dr. for giving me this chance to show our work here. So as introduced in our work, our research focus on method development on LC-MS, metabolomics, and epitranscriptomics. So today I will show our work on epitranscriptomics. So what is transcriptomics? It is a system study of RNA chemical modification in a biological system. So we all know RNA modification plays roles in gene expression. So it plays an important role in various biological processes like differentiation and development. And the recent evidence increasingly highlights the important role of RNA modification in pathogenesis of human diseases like M6A, and GM in liver disease and cancers. Until now, there's one of 117 chemical modifications that identified in biological samples. And you can see from this table, almost all these type of RNA contains chemical modifications. But unfortunately, the functions of these modifications is barely known. Only a few modifications like M6A, M5C was studied. So to know the modification function of these RNAs, first we want to know on which RNA sequence this chemical modification happened, right? But the traditional RNA sequencing method cannot detect these modifications. And during amplification, this information is lost. So people developed the next generation sequencing and direct RNA sequencing. For next generation sequencing, like NM sequencing, ribomethyl sequencing. For this kind of modification identification, for one analysis, and only can identify few modifications. Like in NM modification sequencing, it only can detect AM, UM, CM, and GM. For direct RNA sequencing, basically it's a nanopore technology. For nanopore technology, It can detect various modifications, but this technique cannot discover new modification. And most importantly, these two analyses, this type of analysis is very expensive. For one analysis, for example, for NM sequencing, it costs more than 3,000. LC-MS is a very powerful tool. It can simultaneously detect variable modifications and also can discover new modification. And also, it is pretty cheap compared with this analysis. So we developed a method on LC-MS for RNA modification identification. When analyze RNA modification, we need to digest the RNA sample into nucleoside and analyze this sample in LC-MS for identification and quantification. We also can digest the RNA sequence into oligonucleotide, use different enzymes. If we don't know the modification sequence for these RNAs, we can get RNA sequence from gene bank and use gene bank RNA sequence as database. Use the modified nucleoside here, identified in nucleoside level as a variable modification limitation to do variable modification identification. Also, we can get, if we can find a modified sequence in model mix database, we can use this modified sequence as a database for fixed modification identification, and then map this modified oligonucleotide back to an sequence. So today, my topic is focused on two levels. One is nucleoside level, another is on oligonucleotide level that the method developed in our lab. So on nucleoside level, we detect the RNA modification chains in liver diseases. In alcohol liver disease, we want to know if there is any RNA modification change. We collect urine samples from 18 healthy control and 46 samples from patients with ALD. And these patients we categorize into two groups, non-severe ALD and severe AH use MELD score. After extracting the metabolites from urine sample, we first did an untargeted metabolomics and to use all the significant change in metabolites, did a pathway analysis and see which pathway was significantly changed and what class of metabolites was significantly changed in these urine samples. After that, we did a targeted analysis for confirmation. In the polar profiling on targeted metabolomics, we identified 380 metabolites. Among these metabolites, we identified 25 were nucleoside or bases. 16 of these nucleoside or bases were identified in our in-house database. That means we have their MS spectrum, retain time, and MMZ. That we are confident on this modification identification. Statistical analysis show 10 of these modified nucleoside were significantly changed. Among these significant change metabolites, these 4 show significant increase in the patient's sample. Like in a healthy control, the intensity of this metabolites is low, and it's increased in non-alcohol and getting the highest intensity in severe AH. ROC analysis also show that all these 4 can differentiate non-severe ALD with severe AH. And these 2, 7,9-dimethylguanine and 1-methyl-inosine can differentiate the non-severe ALD with HC, this healthy controls. And you see, especially for 7,9-dimethylguanine, the AUC goes up to 0.9. It's pretty good. Spearman's correlation coefficient analysis show that the 7,9-dimethylguanine is closely related with the severity of the disease. As in the correlation between 7,9-dimethylguanine and the MELD score is up to 0.7 and followed by 1-methyl-inosine, 0.57. And then you can see also from this plot, you can see when the MELD score goes up, the intensity of 79-dimethylguanine is also goes up. Because on technical metabolomics, we submitted all the polymer tablets to the system for detection. So it may have a severe matrix effect. So we want to confirm whether 79-dimethylguanine is very important, is correlated with the disease severity. So we use targeted metabolomics to confirm this result. We get a similar result for 79-dimethylguanine and M22G. And these two can differentiate non-severe from severe AH, and also 79-dimethylguanine also differentiate non-severe ALD with HC. And the AOC, see here, is pretty good. Correlation coefficient also shows that 79-dimethylguanine have the correlation with MELD score as high as 0.66, and followed by MS2T6A and the M22G. So that until now, is to say, in a urine sample, 79-dimethylguanine is really a good marker for ALD disease diagnosis, and also can use for different stage differentiation. So to confirm these results, we also analyzed the alteration or modified nucleoside in serum of these patients. Unfortunately, we did not detect 79-dimethylguanine in serum sample, but we detected MS2T6A, this modification in serum. And we get the same result, similar result as that in urine sample. And also we found these two modifications also increased in severe AH. And all these three can differentiate non-severe LD with severe AH. And the MS2T6A and the acylated cytidine can also differentiate non-severe with HC. And we can say the iron modification may change in ALD, but for severe AH, it's already getting the end stage of ALD. So we want to know if alcohol changed the modification in the early stage. If it is changing the modification early stage of ALD, that means it's possible the iron modification plays some role in disease development, right? So we detected alteration of modified nucleoside in serum of ALD mouse models. And in both mouse models, NIAAA and chronic mouse model, we found certain discovery of modification change in serum. But in serum and urine, the modification change may not only come from liver tissue. It also may come from other tissues like kidney, heart, right? So to further confirm the iron modifications in liver, we extract the free nucleoside from liver sample, and we found 5 modifications were significantly changed in liver sample. We also extracted the total RNA and digested nucleoside. We found the CMUM was significantly changed, significantly increased in NIAAA model, and the CMUM-GM were increased in chronic model along with other 2 modifications. Until now, we can confirm that in RNA level, the modification also changed. So that means iron modification plays some role in ALD disease development. In addition to ALD, we also find a modification change in non-alcohol fatty liver disease. We previously treated the mice with wild Dept, the high-fat Dept. And in the first 12 weeks, we treated the mouse with wild Dept along with vinyl chloride. And we found the vinyl chloride can exacerbate non-alcohol fatty liver disease. We also found that we extract the total RNA digested nucleoside. We found in 12 months, VC only can induce RNA modification changes. And if we compare 12 weeks of VC treatment with the 12 months, we found more modification changes. But this is from total RNA. We don't know this modification from which RNA sequence. So we designed the experiment using the CIEHS treating the human liver cell line. CIEHS is the major, CIEHS is a major toxic product of vinyl chloride. We treat the Hep2 cell line with CIEHS for 4, 8, and 24 hours and extract the total RNA, separate the total RNA into 28, 18S, 5.8 sRNA, and tRNA. We digest these different parts into nucleoside and oligonucleotide for quantification on LC-MS. In 18S RNA, we found 3 modifications were significantly changed. You can say the M70 and M40. At the very beginning, the intensity of these 2 modifications is barely seen. We almost cannot see the peaks. But when after 4 hours of treatment, the peak is increased. The intensity of this peak is increased. And after 8-hour treatment, we can get a statistic significant. HM5C was significantly decreased after 24-hour treatment. And in 28S sRNA, we also find 5 modifications significantly changed. These 3, M6AM and also CM, is significantly decreased. We also found M7G and M4C is increased. We get similar results as that in 18S RNA. Now we know the 18S and 28S RNA have modification changes treated with CIEHS. But we know same modification in different position of the same sequence, they may have different function, like NI modification in messenger RNA. If NI modification happens in 5' of messenger RNA, it will increase translation efficiency with a corresponding gene. But if NI modification happens in the CDS range of the messenger RNA, it may interrupt the translation and lead to the decrease of the corresponding protein. So if you want to know the function of this modification changes, we must know the position of this modification, right? So we want to detect if we want to detect the modification position, we need to quantify the modification quantification in oligonucleotide level. The quantification on nucleoside or oligonucleotide level is quantitative epitranscriptomics. It also has structure epitranscriptomics or interactive epitranscriptomics, but we did not do that. In quantitative epitranscriptomics, the subjects can be RNA. If we detect the modification on small RNA like microRNA, tRNA, we can subject the entire molecules to LC-MS for detection. But for RNA and messenger RNA, it's too large to be detected by mass spec. So I have to digest this RNA into oligonucleotide. Typically, the length of an oligonucleotide digested is from 3 to 15 moles. But it depends on which type of RNAs you use. Like if you use RNT1, T1 digests in 3' of guanosine. If there is a consecutive guanosines in RNA sequence, it's possible this T1 will digest this guanosine into single nucleotide. If it digests into single nucleotide, it will map to everywhere. Especially if we have a modification, we will lose this modification, the position information for this modification. But if we, so when we do oligonucleotide identification, we only use the oligo from 3 to 15 moles. But if we don't know the modification on this RNA sequence, we first need to detect the modification types. So we have to digest the RNA into nucleoside. And into nucleoside, as we previously showed, we don't know, we lost all the position information. So to map the modification back to the RNA sequence, we we need to do the RNA oligonucleotide level identification and quantification. So we developed two platforms for RNA digestion, use RST1 and A. And you can see these peaks were separated very well in these platforms. And about 4,000 features was identified in these two platforms. Now the question is how we process this data, how we know which peak is which oligonucleotide, and this oligonucleotide with modification or not. Currently, there is 4 open source platforms for oligonucleotide identification. Adenine passes, RNA multi-mapper, and the NSE, nucleic acid search engine. So we analyzed our data, use all these 4 platforms. This is the identification result. From the result, we can see adenine identified much more features than all the other platforms. In RST1, it's more than 700 features were identified, and in RSA, more than 850 that identified. And for NIC and PASSIS, about 200 in T1 and 100, more than 100 in A. And RNA multi-mapper identified the least features. And we think if the oligonucleotide identified all these platforms, that we are confident on this identification. So we compared the result of this identification, but we found only two oligonucleotides identified in all these 4 platforms. That's about 1% of the all identified oligonucleotide. And 13.8% of oligonucleotide identified in 3 platforms. And it's even worse for RNA. 0 oligonucleotide was identified in all 4 platforms, and 14.1% oligonucleotide identified in 3 platforms. It's so different. And for modification, you can see from this. For modification, you can see from this heat map, 9 of the modification identified all 4 platforms. What is so different? And which platform is suitable for our complex bulk sample analysis? So for this sample, is bulk sample, we don't know ground truth. So we choose synthesize the oligonucleotide, synthesize the long non-coding RNA, NME1. We treat the NME1 with NCL1. This enzyme will modify the cytidine in RNA sequence into 5-methylcytidine. Theoretically, this enzyme can modify all the cytidine in RNA sequence to M5C, but in reality, we don't know. So we need to use variable modification identification. But for PASSIS, this platform does not have a variable modification function. So we exclude this platform. We compared the identification results in the other 3 platforms, and this is the modification identification result. From this result, we can see our adenine identified much more modified sequence than these 2 platforms. And we also, for NME synthesized, in control group, we did not treat it, the NME use NCL1. That means in control group, they should have no modification identified. But we still see some modification that identified by adenine in the control group. That means it's a false positive. So we calculate the false positive of all these platforms, and we found now in control group, the false positive of adenine is go up to almost 50%. It's pretty high. And in NSE, it's acceptable. The false positive rate is less than 10%. RNA multi-mapper is the false positive rate is 0. But for in the treated group, the adenine identified about 10 oligonucleotide with modification, which is true positive. But the NSE identified about 5, and the RNA mapper identified only 1. That means adenine identified more true positive, but have a high false positive rate. If for biological sample, it's a lot of false positive rate, right? NICE, even though it is have lower false positive rate, but it missed almost half of the for true positive. And for RNA model mapper, it's even lost a lot, even lost the most, right? So for RNA modification, we also can see adenine identified almost every cytidine in this sequence. And only 1 modification identified in all 3 analyses. NSE identified 7 modifications, and 3 of them identified in all 3 analyses, and 2 in 2 analyses. That means it's consistent with this result. So based on this, you can say based on this analysis, these platforms have their disadvantage and advantage. Based on this performance, we developed our own platform, Epiomics. And we use this, we test our platform use synthesized standard, modified standard with different lengths. And we can see here, this is the match result for this standard. The match is pretty good. Almost all the abundant peaks were matched. So next step, we'll test our platform use bulk samples and see how it performs. Now we have the platform for our oligonucleotide identification. Now we move to the RNA modification mapping. Currently, there are several challenges for RNA modification mapping. One is the sequence coverage. People frequently use RST1 and A for digestion. But if you use these 2 enzymes, the sequence coverage is only 50 to 55%. It's half of the sequence information was lost. If we combine RST1, glycine E5, and MZAZF, these 3 enzymes together, we can increase the sequence coverage to 70%. It's a great improvement in this area. Even we use these enzymes, it still may not completely digest. It still have miscleavage. So if we add missed cleavage during data analysis, we can increase the sequence coverage to 80%. But still, it have 20 to 30% of the sequence were missed. The second challenge is the confidence on the modification identification. If we use this enzyme to digest, we know this enzyme have base specificity or sequence specificity. So typically for one sequence, this enzyme digests only create 1 oligonucleotide. If even have miscleavage for some site, some position, it only have 2 or 3. What if we identify the modification in these 2 different 2 oligonucleotides? They are different. Which one is right? It's 50-50, right? So we are thinking if there is any way that we can simultaneously increase the sequence coverage and the confidence of modification identification. In 2022, there's a publication on environmental science and toxicology. The authors established the characteristics of metal ion digestion to the RNA backbone. Metal ion can digest in the 3' of any basis. There's no base specificity and no sequence specificity. So I'm thinking whether we can use this method if in one sequence, in one digestion, this metal ion may digest the same position to different oligos. And if this is the case, it may increase the sequence coverage. And if this modification, this oligos they have modification when identified most of the modification as the same modification. Then you also increase the confidence of modification identification. So we tried metal ion, we choose zinc to digest E. coli RNA. And this is the identification result. In zinc digest, we identified much more unique oligos than compared with RNA and T1. And the oligo length in the zinc digest is also longer. The average length of the oligo is 15 mers. Compared with the RNA and T1 is about 9 mers. So if the length of the oligonucleotide is longer, that's increase the possibility to match the oligonucleotide of a single position of a single RNA. So we analyzed the three analysis of this zinc digest. We compared the result of these three analyses, and we found the reproducibility of zinc digest is very poor. It's only 9%. That means we cannot use this method for oligonucleotide quantification, right? But for identification, this is what we expected. Let's see this result. In zinc digest, in E. coli RNA, in this position, it have M7G modification. And this position was created in zinc digest 11 oligonucleotide, these different oligos. And all these oligos identified as M7G modification. Then we are very confident on the modification identification in this position. And well, in RNA and T1 even have miscleavage. It is in 3 oligos or even only 1 oligo. So for the modification, compared with these 2, we are more confident on the zinc digest products. And for the sequence coverage, zinc digest can increase the sequence coverage for both large RNA, small RNA, up to 96%. And for RNA-T1 and RNA, it's about 54, 56%. And you can see this green background. This is the missed information for this sequence. For the modification identification, it's the same. Like in zinc digest, most of the modification identified in all 3 analyses. But for RNA and RNA-T1 digest, many of the modification only identified in 1 or 2 analyses. So from this data, we can say that zinc digest can provide more informative oligonucleotide. And this oligonucleotide increase the sequence coverage of the RNA to 96%. And also, if identify the modification in multiple different oligos for the same position, also can increase the identification confidence. But the drawback of this method is the poor reproducibility of zinc digest. We cannot use this method for quantification. We only can use the method for identification. So, except for this method developed in our lab, we also develop a method on identification and quantification of glycan that's released from RNA. The reproducibility of this method is pretty good. It's up to 70%. Use this method, we identified more than 200 glycans in the total RNA that extracted from mouse liver. And then using method, we also can identify the composition of these glycans. This data is submitted to analytical chemistry, and now it is under review. Next step, we will develop the method. We will map this glycans modification to the RNA sequence. We will do the modification and the quantification in the oligonucleotide level. And we also developed method for RNA modification quantification on oligonucleotide level. And for the small RNA, microRNA, tRNA, we will develop a top-down epitranscriptomics. And we already have get some data for this too. Okay, this on the presentation for today. I want to thank my supervisor, Dr. Xiang Zhang, designed all this experiment and supervised all this experiment. And our collaborators like Professor Mueller, Eugene Miller, also provided very valuable suggestions. This Audrey Dept is the PhD student working on zinc digest, and Fengjing is working on the platform analysis, epigenomics analysis, and Carol is the student working on the glycan modification, RNA glycan modification. Regina and the students work on top-down analysis. I also want to thank to the NIH grants to support our research.